Good afternoon. Can you hear me? Hello. 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 Navya. Hello, Navya. Hello. Can you hear me? Navya. Hello. 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 అమ్మా ఎవరైనా వినబడుతుందా? ఎవరున్నారు ఆన్లైన్ లో? హలో. రాకేష్ గారు. హలో. రాకేష్ గారు. హలో. హలో. హలో నవ్య. హలో నవ్య. హలో. హలో.
గుడ్ ఆఫ్టర్నూన్ సార్.
ఏమ్మా వినబడట్లేదా?
అవును సార్, ఇప్పుడు వినిపిస్తుంది సార్.
ఎందుకు ఏమన్నా పవర్ ఫెయిల్యూరా?
ఎస్ సార్.
అవునా?
ఎస్ సార్.
నవ్య కదా, ఫ్రమ్ నిజామాబాద్?
కాదు సార్, మాది పొదిలి. నవ్య శోభ.
ఓ నవ్య శోభ. అచ్చా, లాస్ట్ టైం ఇంకొక అమ్మాయి నిజామాబాద్ నుంచి, ఫ్రమ్ పొదిలి, ప్రకాష్.
ఎస్ సార్.
ఓకే, మంచిదమ్మా. షల్ వి బిగిన్?
ఎస్ సార్.
బికాస్ ఇట్ ఈస్ గెటింగ్ లేట్, యు నో.
ఎస్ సార్. ఓకే సార్.
Is this the first class for you? Were you present in the last 2 classes?
నేను లాస్ట్ 2 క్లాసెస్ కూడా వచ్చాను సార్.
వచ్చావా? ఓకే ఓకే. మరి బిగిన్ చేద్దామమ్మా, టైం అయిపోతుంది కాబట్టి.
చేద్దాం సార్.
రైట్. ఓకే. సో as you know, in the last 2 classes we discussed about the ultrastructure of the cell organelles, And in other class, in the last class, we discussed about the genetic material, that is nucleic acids, that is structure of DNA, structure of RNA, and their types and the method of replication, etc. We studied in our last classes. Now today, what we do is, we'll try to learn about the cell division. Today, in today's class, we discuss about the cell division, types of cell divisions, and also the extranuclear genetic material. So these are the two topics we would like to cover in the next almost 65 minutes. Okay, so to begin with, cell division. Now, this is not the first time for you because this topic you learn from your high school onwards. I think cell division is there even in 8th, 9th class. It is being introduced for the high school students. And of course, you read about this cell division in detail in your intermediate. Now, what I advise you is, so before you study your material that is being provided to you by Open University, my sincere advice you is, you try to go through the intermediate syllabus so that you can get the basic information from that. Whatever the little bit extra that is being added in your course, you will be in a position to understand a bit more, and you will be in a position to attempt your examination with all confidence. Okay, with this background, let us begin with cell division. As you know, cell is the unit of structure, cell is the unit of function. This is according to cell theory given by Schleiden and Schwann. Now, other man, that is Rudolf Virchow, another man who in 1859, he added a point to that cell theory. He said that always new cells arise from the pre-existing cells. There is no scope for de novo formation of any new cell. This, he said, omnis cellula e cellula, that is always new cells arise from the pre-existing cells. This is the meaning of omnis cellula e cellula. Now, every life form, starting from that of fishes, amphibians, reptiles, mammals, or from cryptogams to phanerogams. You take life cycle of any organism, you'll be amazed to know that the life begins as a single cell. Single cell is the starting point, even including human being. When we are in our mother's womb, we are just single cell, a zygote. And this zygote develops. Development is here. What do you mean by development? Here, the cell, the starting cell or the zygote undergoes repeated divisions. As a result of that, you will get hundreds, thousands, millions, and billions of cells. And these cells, organize themselves into tissues, and in turn, they give rise to tissue systems, organs, and ultimately a new organism or a new individual as such. Now, even if you take starting from your childhood, when we were kids, when we were 1 month old, when we were just born, very small, and as we put in time, now our body grows very fast. Up to teenage, we grow faster and faster. Thereafter, of course, growth in elongation stops, but of course, we add in width. We increase our weight. And of course, this is a lifelong process. This is a lifelong process. The cell division never ends as such. Because you may ask me, how this, sir, when we get old, generally we don't grow. Some people, neither they show any increase in height or weight. But if any injury occurs, if you observe that, that injury gets cured. It is because of formation of new cells. So new cells are being added. See our skin and this epidermis, this dermis, these cells have the ability of undergoing repeated divisions. So it is because of division. It is because of cell division, new cells are added and growth and development results. So now this is the cell division. Now cell division is very common process, very common phenomenon, starting from unicellular organisms to multicellular organisms. But in unicellular organisms, cell division means it is a reproduction. Cell division means reproduction because one cell becomes two cells. If you take chlamydomonas, if you take paramecium, if you take a bacterial cell, there they undergo division. Maybe a binary fission or it could be a transverse, longitudinal, and in any plane as such, whatever the case may be. So new cells are being added. So in unicellular, 1 becomes 2, 2 becomes 4, 4 becomes. If you take a bacterium, and for example, if you take a Chlorobacterium, that is Cholera vibrio, it divides, within 24 hours. Within 24 hours, millions of cells are produced, and that is because of, if cholera breaks, the entire village gets affected, and not only your village, but even your surroundings get affected because the excreta that is being added to your water bodies, which is contaminated. If you happen to take this contaminated water, you become indisposed and you are subjected to this cholera disease. So only this cholera bacterium spreads, it is because of a very fast division. It's a new daughter cell. A daughter cell within 30 minutes, it attains, a maximum size and it is ready to undergo division. So it is because of cell division, new cells are being added, and this is what we call, either growth and development or a reproduction, as the case may be. Now we move to, now what do you mean by a cell cycle? Cell cycle, for example, if a cell divides, you get two daughter cells, but the daughter cell cannot immediately undergo division. Before that, what it has to do? It has to grow. It has to attain maturity, adulthood, full, structure. Then only when it is fully equipped, fully grown, then the cell can enter into division, and again it divides and produces new cells. Now, a daughter cell, it grows, it expands, it gets its genome gets duplicated, then it is ready. So then it undergoes division and produces two. So this series of events starting from a daughter cell, which increases its size, increases its protoplasm, increases its DNA content, increases its cell organelles. Then when it attains its adulthood, when it reaches a stage, at a particular stage, then it is ready to undergo division. So these series of division, these series of events, starting from a young daughter cell to its adulthood and it undergoing again division, this series of events we call cell cycle. Now, the division as such, taking or speaking about the cell division, the cell division, divisions are of primarily of three major types, direct cell division or direct division, indirect division, and the meiosis. So direct, what do you mean by direct cell division? This direct cell division is observed in primitive organisms or generally prokaryotes. Take an example of a bacterium. Here, the bacterium cell very small, microns in size, and when it grows fully, when it is ready to enter into a division, here, of course, cell is there. It is enveloped by a cell wall, and you have what we call a plasma membrane. And inside that, what you have the centroplasm, that is DNA region. Here, of course, because in prokaryotes, you don't find a nuclei or membrane, and the genetic material is not organized into a distinct chromosomes. Why? Because there is no histone protein. Because of the absence of protein, histone protein, the genetic material is what we call a naked DNA. And this DNA, this DNA gets replicated. This DNA gets replicated. So now here what happens? You don't find chromosomes, and generally because it is not bounded by any membrane. Now the nuclear region gets elongated. Then it roughly divides into two. With the invagination of the cell wall and the cell membrane, it ultimately divides. For example, if it is the nuclear region, if it is like this, now when it enters into division, here it happens like this. Ultimately divides into two parts. And because there is no involvement of chromosomes, or chromosomes are not formed, no nuclear membrane. So here the nuclear material or the cytoplasm divides into two and is followed by division of the. So when it invaginates, it not only divides the cell organelles, but of course when you speak of in a prokaryote, generally you find only ribosomes. Other than that, no organelle is there. So other materials also get equally distributed, and you get ultimately two cells. Now these cells, again, they grow, they attain the adulthood. Of course, they again enter into division. So they a kind of division that where no nuclear membrane is not formed, nucleolus is not formed, and cell organelles are not formed, and the DNA ring roughly, it divides into two, and such mode of division is known as direct division. Then indirect division. Now indirect division is a division, a cell division wherein the changes occur not only in the nucleus but also in the cytoplasm. Now here, because a series of changes the chromosomes or the nucleus undergoes, and ultimately you get two nuclei, and each nucleus gets migrated into the first daughter cell, and thus at the end of the division you get either 2 in case of mitosis or 4 in meiosis. So now here in this indirect division, first of all, the division of nucleus takes place and is followed by the division of the cytoplasm. Division of the nucleus is known as karyokinesis, and the division of the cytoplasm is known as cytokinesis. So karyokinesis is followed by cytokinesis. In some cases, if cytokinesis fails, if the nucleus undergoes repeated divisions, but there is no separation, then that results into what we call multinucleate condition, otherwise known as a sinusotic condition, which is the characteristic feature of generally fungi. But of course, in angiosperms, in certain cases, especially in latissiferous tissue or in tapetal cells, you find more than one nucleus. So now we will, first of all, we will try to learn about, before discussing about the various stages, various phases of the division, let us learn a bit about the cell cycle. I already told you, it is a cell cycle is a phase wherein series of events take place. By the end of that, a small daughter cell, it gets or it reaches an adulthood stage wherein it can enter into division phase. So for our convenience, we divide cell division into two important processes. That is, one is the interphase, the other is mitotic phase. Interphase is the first phase, but of course, interphase, previously people used to have a misconception. They used to think that in interphase nothing happens, and interphase is a resting phase as such. But it is wrong to say a cell that is in interphase is taking rest, but it is very active. It prepares itself for the division. A number of events take place in the interphase phase of the cell, wherein it grows and it multiplies its genome, that is DNA gets replicated, and all cell organelles also, they get they grow and they get themselves, distributed, or they to multiply. So there is a replication of DNA and also increase in the number of the various cell organelles, so that an adult well-equipped cell results, and then it enters into division phase. So this, this preparatory stage. So interphase is a preparatory phase which prepares itself to enter into next phase of division. So now interphase, for our convenience, we divide this interphase into three subphases. One is G1 phase, S phase, and G2. G means here gap. G1 is the gap phase, first phase, wherein what happens, here the cell expands and there is an increase in RNA content, there is an increase in protein content. So, this cell, after G1, it enters into next phase, that is S phase. S phase, during S phase, the major change that occurs in S phase, S means synthetic phase. What is known as synthesis? Synthesis of DNA takes place, or duplication of the DNA takes place, or the replication of the DNA takes place in the S phase. So S phase is the most important phase, most active phase of the interphase, or or preparatory phase of the cell division. And after synthesis, then what happens? We enter, or the cell enters into what we call G2 phase. During this G2 phase, then there is an increase in RNA. There is increase in enzyme content. There is increase in protein content. So proteins, your carbohydrates and other important materials are produced in sufficient quantity. Now the cell reaches a stage, or reaches an adulthood stage, so that it can enter into division phase. So first phase, a very active phase, is what we call interphase, during which cell prepares itself. Cell itself reaches a stage wherein it is ready to enter into M phase. M phase is known as mitotic phase. So now I told you G1, S, and G2. So if you assume that for the cell division, a cell takes around 24 hours, means the cell spends around 20 hours in interphase only. Even more than 20 to 22 hours, it remains in the interphase, or it prepares itself in this longer time span. Then the division phase is very short. It lasts for only 2 to 3 hours. So for maximum time, a cell spends, or cell prepares itself for division phase. So a cell has spent a lot of time during G1, S, and G2, which lasts for about 80 or 90% of the time. Then it is ready to enter into division phase. That is, next phase is what we call M phase, or mitotic phase. So this is the division phase. Now this division phase, it very short, and you have to remember, cell never stops. There are no commas or full stops. Cell, the changes are continuous. Say, for example, a cell is in G1 phase, means there are no commas. It doesn't stop there. It's a continuous process. We don't know. When it is complete, then automatically it enters into synthesis phase. There, the DNA replication starts. It ends. Then it enters into what we call G2 phase, and ultimately it enters into mitotic phase, or division phase. Now this mitotic phase, or the division phase, this division phase, I told you, very short. And this division phase, for our convenience, this division phase, we divide divisions into two major types. One is mitosis, the other is meiosis. So mitosis means here, why it's known as mitosis? Here, when a cell undergoes division, it produces its two daughter cells, which are very, very similar to its mother cell in all respects, except in size. Except in size, they share various features of its mother cell, including the most important component is chromosomes. So the chromosome number is being retained. If there are 10 chromosomes in mother cell, daughter cells do have 10 each. So only thing is, when compared to the mother cell, the young daughter cells are very small. Except that, in all respects, it is very, very similar to its mother cell. Of course, size is less, the cytoplasm is less, but even various cell organelles are very small, and within no time, they grow, they attain full, full size. Then, of course, the cell again becomes ready for division. So for our, what I want to stress here is, cell division means various phases are there. There are no commas and full stops. It's a continuous process. So for our convenience, for the convenience of the students, for the convenience of the beginners, scientists, they divided this into a number of subphases or stages based on the important changes that are taking place in chromosomes or the nucleus. Now, say, for example, let us begin with mitosis. I told you mitosis is otherwise known as equational division. Why is it not equational? As a result of, as at the end of that, two daughter cells which are very, very similar to mother cell. Chromosome number is retained. And this generally occurs in vegetative cells. In vegetative cells, opposite to vegetative cell is reproductive cells, sex cells. So mostly in all vegetative areas, vegetative cells, tissues, this mitosis occurs. So now the other important thing is when there is an injury, repair, tear and wear and tear, in such cases also new cells are produced, means these cells are produced as a result of mitosis only. So mitosis helps in the retention of the chromosome number. It is needed for growth and development. It also helps in reproduction. It helps in wear and tear. So it takes place in haploid cells and also in diploid cells. It is a very common and most frequently occurring mode of division is mitosis. So based on the behavior of the chromosomes, based on the changes that you observe in the cell division, this phase is divided into, of course, these are very well known to you: prophase, metaphase, anaphase, and telophase. Prophase is the first phase. So here what happens, I'll stress only on the important points. So in prophase, what happens? The nuclear membrane is lost, nucleolus is lost, and the chromosomes, which are in the form of a network, they tend to coil, they reduce their length and become very thick, become very short. So initially you find a chromatin network, chromatin net, but later on, as the time, as the cell prepares, as the cell moves, then the coiling takes place, and the nuclei or the network ultimately gets because of coiling, because of its length is reduced, and you will see small rod-like structures under a microscope, and then you start calling them as chromosome, colored thread-like structures. So coiling takes place, length is reduced, and this we call condensation. Condensation of the chromatin occurs. Chromatin material occurs, and ultimately you get chromosomes. And at the end, the nucleolus is lost and a nuclear membrane. Then, because when the envelope, that is, when that is the layer that is delimiting, restricting the movement of chromosomes, and when it gets disorganized, they become free. They become free and come and occupy the center place of the cell. This we call metaphase. After prophase, metaphase comes. During metaphase, all the chromosomes come to the equatorial plane, and they are there on the equator of the cell, and very short very condensed, clearly visible. And this is the stage which is most convenient to know the details of the chromosome. So which is the, under objective, they may ask, which is the stage that is most suitable to know the structure of the chromosomes? It is the metaphase. So then comes telophase. So prophase, metaphase, and telophase. When the telophase, when the cell enters into telophase, what happens? From each pole, you will see spindle fibers, origin of spindle fibers. Now spindle fibers are like this. Spindle fibers, they originate from two poles, and ultimately, of course, these fibers, spindle fibers, they attach to the primary constriction, otherwise known as centromere. So they're from other side, from two sides, the spindle fiber comes and attaches itself to the kinetochore or the centromere area. Now this, you will see what we call formation of the spindle fibers. And anaphase, what happens? What is the purpose of these spindle fibers? They tend to pull the chromosome from one side, from the two sides. When chromosome is like this, they are pulled apart. So as a result of that, when the chromosome, by the metaphase, you will see chromosome splits longitudinally. In each chromosome, you find what we call, initially it is like this. But as the condensation increases, it shows two chromatids. Two chromatids are like this. Two chromatids are the constriction of the centromere. Now, because of the pulling, what happens? Because the pulling, these chromatids, they move. The two chromatids are daughter chromatids, daughter chromosomes as such. They move to respective poles. And then comes your telophase, or the last phase. Now, during this phase, whatever the changes that occurred in prophase just occur in reverse. Now, they decondensation. In prophase, what was there? Condensation of the chromatin. Here, decondensation. Condensation takes place, and again, reformation of what we call chromatin material, and the reappearance of nuclear membrane and the nucleolus occurs. Now, as a result of that, in a cell, at the two poles, you find two rounded bodies. They are nothing but your two nuclei. Now then, this is, so nucleus got divided into two nucleoli. Two daughter nuclei are produced. Then comes what we call division of the cytoplasm. Not only nucleus, but even the cell organelles and the cell material has to be distributed equally. This division of the cytoplasm, which we call cytokinesis. Cyto means cytoplasm. So division of the cytoplasm takes place, and this for the division of the cytoplasm, what happens? Whatever the spindle fibers that were there, so their purpose ends by pulling the chromatids to respective poles. Now, what are the remnants of the tubules, or the remnants of the fibers? They organize into, in the center you will find like this, a cylindrical structure, which we call Fragmoblast, and the all microtubules or the vesicles that are generated on the endoplasmic reticulum or on the Golgi, they form a small thin layer in the center of the cell, and ultimately this develops into plasmodesmata, and later on, with the addition of the cell wall material, formation of or the division of the cell occurs, and ultimately you get two daughter cells. So this division, what we call cytokinesis. So karyokinesis and cytokinesis. So to repeat, the purpose of the this is the, so they may ask you, give or write briefly about mitosis with illustrations, and add a note on its significance. So its significance is to maintain the constancy of the chromosomes, and for the growth and development. It occurs in all types of cells, maybe haploid or diploid, and for wear and tear, it helps. And it is because of this, the growth and the growth, which is what we call a permanent change. A permanent change. So growth is a permanent change. So it's because of that growth and development takes place, reproduction takes place, and retention or maintenance of the constants in the chromosome number is there. And for injury, recovery, or the reformation of or production of new cells is because of this mitosis. Now, this is the significance of mitosis. Then we move to what we call second more type of division, that is meiosis. Now, this meiosis is, this occurs. It differs from the mitosis. It is otherwise known as, meiosis is otherwise reduction division. Why is it known as reduction division? That was known as equational division. Why? Because two daughter cells are produced which are very, very similar to its mother cells in all respects. That's why equational. Here it is reduction division. What reduction takes place here? Reduction in the chromosome number. If the mother cell has 10 chromosomes, the daughter cell shall have 5 each. So as the chromosome number is reduced to half, that's why it is known as reduction division. So generally, unlike mitosis, which is vegetative, which occurs in all types of cells, but reduction division is restricted only to germ cells or reproductive cells. Maybe your microspore mother cell or a megaspore mother cell. So only in sex cells, this reduction division takes place, and this takes place only in adult well-matured cells. Only in well-matured cells, the reduction division takes place. As it is, at the end, what do you get? You get gametes. Gametes. So for when the cell enters, or when an organism enters into, when it is ready for sexual reproduction in the sex cells, in the germ cells, generally the reduction division takes place. As a result of that, you get formation of the gametes, which have half of the chromosome number of the mother cells. So now let us know about this meiosis. Now meiosis, for example, it differs from mitosis. As a result of the mitosis, you get only 2 cells. But whereas in meiosis, at the end of the meiosis, you get 4 cells in general. So here division occurs twice. Here the division occurs twice. So to differentiate, what we write what we call meiosis 1, Roman 1, and meiosis 2. So these 2 divisions occur successively. So in the meiosis 1, what happens? The mother cell undergoes. As a result of that, you get 2 daughter cells. They are known as dyads. They have half of the genome, haploid set only. So, and these are 2 daughter cells or dyads, and these 2 dyads, they again, without any gap, they again enter into division, and from each daughter cell you get 2 more daughter cells, and this. So at the end you get 4 cells. So that's why it differs from mitosis, where only 2 cells are produced. Here at the end of the meiosis, you get 4 cells in general, and chromosome number is just half. Now meiosis 1 and meiosis 2. So meiosis 1 is very, very important, very, very complicated. Let us try to understand this meiosis 1. So meiosis 1 also, here also there are 4 phases similar to your mitosis. First phase, what we call prophase, metaphase, anaphase, telophase. Only 4 phases are there. But the prophase, very lengthy. Prophase is very lengthy, very complicated, very, very important. Now prophase, similar to that. What happens at the end of the prophase there? At the end of the prophase, there is appearance of the nuclear membrane, losing of what we call nucleolus, and the chromosomes are set free. They come and occupy the center of the cell, equatorial plane of the cell. So the same. At the end of the, here also in meiosis also, disappearance of nucleolus and nuclear membrane, and the chromosomes are set free. But before that, lot number of changes do occur in chromosomes. So let us know about that. So in meiosis 1, prophase is for our convenience based on the changes again. So we give importance to the changes that take place in the nucleus or in the chromosomes. So here, prophase, for our convenience, we divide this prophase into 5 subphases. 5 subphases. First one is leptotene, zygotene, pachytene, diplotene, diakinesis. 5 subphases are there in the prophase 1. Now, first is leptotene. Leptotene, and the name leptotene means, if you observe the cell, the mother cell that is ready to undergo division, if you observe the nucleus, the nuclear material or the chromosomes are in the form of small threads, lengthy threads. So lengthy colored threads are seen in the metaphase, prophase 1 of the meiosis, especially in the leptotene. So long lengthy structures, very coiled, thin structures with many chromomere-like structures are there on the chromatin. Then, so here also what happens? The condensation of the chromatin takes place, or chromosome fiber takes place. And of course, you will see the formation of small rod-like structures appearing. Of course, they may be what we call, they may undergo further condensation by the time of your metaphase. So here, what happens in the leptotene? Very thin structures are there. When they enter into zygotene, what happens? Pairing of chromosomes takes place. We are diploid. We have billions of cells which are diploid in nature. Diploid means two sets of chromosomes. You get one set from your father, we get one set from our mother. Two sets are there. So you and me have what we call 46 chromosomes, 23 pairs. 23 pairs you get from your father, 23 you get from your mother. So in all 46, 23 pairs. Now here what happens when the cell is in zygotene, the pairing of the homologous chromosomes takes place. Pairing of homologous. Homologous means one from father, the other from mother. Pairing. When the pairing takes place, what do you get? We call such pairs bivalents. For example, if there are 10 chromosomes, let us assume you get 5 bivalents. 5 bivalents. 2, 5 bivalents like this. This 5, this 5. They form what we call 5 bivalents. They are there. Some are small, some are big. And this what we call dyads are bivalents during zygotene. Now during this zygotene, what happens? This pairing is helped by what we call synaptonymal complex. A number of enzymes, a number of proteins help in the pairing of the chromosomes. In the pairing, what happens? They twist one another, around one another, very close. This we call pairing. As a result of pairing, you get bivalents. So, zygotene means pairing or synapses, otherwise known as synapses, helped by synaptonymal complexes. This is zygotene. Then pachytene, next phase, next subphase. Pachytene means here, if you observe a chromosome initially, it will be like this. When zygotene, 2 chromosomes are like this, pairing, one father, one from the other mother, 2 pairing. Bivalent we call. Bi means 2 chromosomes pairing. Then when the cell enters into pachytene, further condensation, further shortening of the chromosome takes place, and the chromosome, with the shortening, you see, in each chromosome, you will be in a position to find what we call chromatids, 2 chromatids. Initially, they are like this. Initially, these 2. But with further condensation, what do you see? With further condensation, we will see what we call, in each bivalent, you find 4 chromatids. One chromosome, the other chromosome. 4 chromatids. As there are 4 chromatids, this we call what we call pachytene tetrad. Tetra means 4. So in packaging stage, you find in each bivalent, you find 4 chromatids. This we call a packaging. Now this packaging tetrad, now here if you observe further, what happens here, the chromosomes, not only they got paired, synapsed, and you will see what we call with the overlapping, twisting of one chromatid around the other. And these generally, what we call the chromatids of each chromosome are known as sister chromatids. Sister chromatids. This is a sister chromatid. Now, what is the relation between these two? These two are sister, these two are sisters. This and this, non-sister. These two are non-sister chromatids. So non-sister chromatids tend to pair with each other, twist around each other. And at the places where they are twisting, getting twisted, because of this tension, they break. At the areas where they got twisted with one another, breakage of the chromatid takes place, and then exchange of this material with that, and taking that part of the chromosome to this takes place. This we call what we call exchange or crossing over. Now at the places where they are touching, there they break, and there the exchange of the chromatid takes place. This we call crossing over. And at the crossed over regions, so where are we now? We are in the packaging. Then the cell enters into diplotin. At the diplotin, what happens? So till now, in the zygotine and packaging, what was there? There was a pairing. There was an attraction between the non-sister chromatids. Now, after the exchange, what happens? These sister chromatids, they tend to repulse from each other. They tend to move away from each other. The exchange is over. They don't want to remain together any longer. They want to separate. They want to now they want to separate. With this, what happens? The X structures, what we call X. So this is an X structure, X-like structure, which represents the spot where the breakage of the chromatid has taken place, and the where exchange of the chromatids has taken place. And there, at that point, you will see this X-like structures. And these X-like structures are known chiasmata. With the repulsion, what happens? With the repulsion, with the moving away, this X or the chiasmata, they tend to sliding. From the center, they tend to slide to the tips. This we call terminalization. Movement of the chiasma from the center to the tip of the chromosome, what we call terminalization. The terminalization takes place in the next stage, that is what we call diakinesis. So leptotin pairing, packaging 4, then formation of chiasma and crossing over, then movement of this chiasma to the tip, this we call terminalization. So with the terminalization, what happens? The exchange is completed, and they are repulsing from each other. And this brings an end to what we call prophase. Then rest of the phases are very similar. Now, by this time, what happens? Nuclear membrane disappears, nucleolus disappears, and all these chromosomes are set free. They come and occupy the center of the equatorial plate of the cell. Now, this phase, that is positioning of the chromosomes in the center of the cell. This we call metaphase or metaphase plate center. Then at this point, what you see? Same as like in mitosis, formation of what we call threads, chromatin threads, or what we call the spindle fibers. The spindle fiber or spindle apparatus, formation of the spindle apparatus begins starting from two ends, both ends. Now they come like this, and from one thread from this pole and the other thread from below, and they come and join onto a chromosome. Now anaphase means pulling of the, so what are there? The chromatid pairs are there. So one pair from the mother, other pair from father. Now they tend to separate. They move to respective poles. This we call telophase, movement of the homologous chromosomes, or the two bivalent tend to separate, movement of the chromosomes. Now, one point you have to concentrate here is, in case of mitosis, there was the movement of chromatids. But each chromosome got longitudinally divided, and you find two chromatids, and two daughter chromatids, what we call daughter chromosomes, they got separated. But here in meiosis, what is happening? There is a movement of entire chromosome. One chromosome moves to the one center, the other chromosome move to the other pole. Movement of the respective chromosomes to the opposite poles. This we call anaphase movement. Now during the anaphase, what happens? If you observe a chromosome, in case of your mitosis, or here also, chromosomes appear either a shape of a V or L or I. If it is a metaphase, generally it will be in the form of V. If it is acrocentric, one arm is very short, the other arm is very lengthy. Acrocentric. Telocentric, it is I, small I shape. The centromere is at a very extreme tip. So the shape of the chromosomes are observed well when the cell is in the anaphase. Then, of course, telophase. Then, similarly, reformation of your nuclear membrane, nucleus, etc., takes place, and the division of the cytokinesis, that is division of the cell. So at the end, you got two daughter cells, and these daughter cells have half of the chromosomes of mother cell, only 50-50. Now these daughter cells, they immediately enter into the next phase, that is meiosis 2, which is very, very similar to your mitosis. Here, each chromosome splits, and two daughter cells. From this dyad, you get two daughter cells. From that dyad, you get two daughter cells. And ultimately, at the end of the meiosis 2, you get 4 daughter cells. This is what we call meiosis. Now, what is the significance here? Significance, very, very important. They may ask you, describe the process of meiosis with suitable diagrams and add a note on its significance. Very, very important. So here, the significance is reduction in the chromosome number. Say, for example, if there is no reduction in the chromosome number, what happens? If the sperm and the egg meet, say we have 46. If there is no reduction, 46, it leads to what we call 92. If a cell has 92, it never looks like its mother cell. At each generation, a new species or a different organism will arise, and within a short span of time, the number of chromosome gets doubled, tripled, and multiple number of chromosomes takes place. And because of that, a series of repercussions will be there, and the organism never resembles its mother. So to avoid that, meiosis is very important. For the maintenance of the constancy, this is very, very important, because further we have 46. We have 46. So half from the mother, half from the father. 46 means your sperms will have 23 chromosomes. Mother's egg will have 23. These 23 fuse, and your zygote results, and that zygote has what we call 46 only. So that's why offspring or the children resemble their mother, because the number of chromosomes is retained. The constants of the chromosome number is maintained only because of this meiosis. And what is the significance? Say we have seen in leptotene, zygotene, packaging, what happened? There is synapses and there is exchange of genetic material. With that, we have what we call recombinants. The recombination, variance is taking place. So the variations and the mutations, of course, mutation is not the here. So here, because of the variance, exchange of genetic material resulting into what we call variance. And whatever the small, differences we exhibit. See, for example, you and me, we both belong to what we call Homo sapiens. You have 46, I have 46. But I resemble my parents and you resemble your parents. And I look like my brothers. I look like my sister. My sisters are equal. I mean, they share many features. And my brothers share many features because we are born to same parents. But still, me and my brother have distinct features. These distinctive features are specific to each individual. So there are more than 650 or 700 crores of population all over the world, but no two individuals are perfectly same. It's very impossible. Why? Because cell undergoes hundreds and thousands and lakhs of divisions. During these divisions, there is every possibility for small change to appear. And these small changes are sufficient to bring a small change in your phenotype, in your expression, your characters, the way you walk, the way your importance, your behavior, etc. So our behavior, our individuality, our personality is a product of your heredity and environment. Your heredity and environment, there is a constant, interaction between heredity and environment, and ultimately your personality is because of this interaction of your heredity and environment. Say, but for example, if both brothers, siblings, if they are grown, if they are reared in different areas, maybe your environment has effect. So two offsprings, one is develops in Kashmir-like area, the other in Tamil Nadu-like area. But of course, they have same features, but their external features will be different. A person growing in Kashmir generally because of cold environment, fairer. But a person in South, where the temperature is more, generally you have tan, that is black. This so in genetically not much difference, but because of environment you will see. But so also your peer groups, the school where you are educated, and your peer, your teachers, your neighbors, etc., exert some pressure or the as other, and that determines your personality. So this is about mitosis and meiosis. Then apart from this, we'll turn to learn something about what we call the regulation. The regulation of the division, cell division, regulation of the cell division is what we call checkpoints here. See, at number of, I told you a cell starting from your zygote till adulthood, the zygote underwent million number of times cell division has taken place. So such a large number of cell divisions do take place. There is every possibility for some or the other injury, some or other deviation tend to occur. But in the cell, in our genetic complement, there are various processes that check your deviations. So now they are called what we call regulation and checkpoints. Say, for example, like this. For example, if a vehicle is coming from RTC garage, there the mechanic checks its brakes. It's one checks a tire, the air pressure, etc., will be there, and grease, etc., petrol, etc., will be checked. Brakes will be checked. Then it passes out, and one stage is over. Then the other security man checks whether all checks all the parts, important parts, which are glasses, etc., are there, seats, etc., condition. Then it comes out. So if they find any deviance, any shortcoming there and there itself, they check it and they correct it. Then only it comes out. Here also, when the cell enters into division phase, we have seen there in the first or the G1 phase or the interphase, there we have seen at G1. So various, various metal ions are there, various enzymes are there, various proteins are there. They check. If any deviance is there, they try to correct it. So unless and until it passes G1 check, it can't enter into what we call S phase. Unless and until it clears signal from the S phase, it then enters into what we call G2 phase. So at every stage, there are checks and balances. If there are no checks and balances, there is every possibility for the formation of an abnormal cell. Abnormal cells may result, and that abnormality may proliferate, and you will see formation of warts, etc. Say, for example, our cancer is nothing but an altered cell division. Because of altered cell division, some error has taken place. Because of that, you got a cell, a maligned cell, a modified cell, and that cell, unless and until it is checked, it spreads, it divides and redivides. Without any control, uncontrolled division. Cancer is nothing but uncontrolled division leading to the formation of lump of cells. So such deviations are checked by what we call the checking points. So checking points are there at the G1 phase, S phase, and G2 phases. So now these checks, these checks are at the gene level, or at what we call it protein level, or at enzyme levels, especially what we call molecular regulation. Molecular, at the molecule level, regulation takes place. And this has, these are checked by what we call, there are two important molecules that are involved in checking these things. One is what we call cyclins. They are nothing but proteins. The other is CDK, that is the cyclin-dependent kinase. CDK means cyclin-dependent kinase. So cyclins as such, they are proteins. They are not active alone. Unless and until they meet this CDK enzyme, then they become active. And for their joining, for these two proteins to join and to become active, what is needed? ATP is needed. So you need what we call cyclins, you need CDK enzymes, and you need ATP. Then only they become active. At the active stage only, they check. They check whether the cell that is passing from what we call G1 can enter into S phase, or a cell which is in the S phase, can it proceed to G2 phase. So like this, at every stage, there are checks and balances at the micro level. At the very micro level, these checkings are there. Because of that only, though we have billions of cells, which our zygote has undergone in our mother's womb, so it underwent lakhs and millions of times cell division, but errors are very, very negligible. Very nil errors have taken place. So that's why we are normal. We look like our parents, our personality, and in many features, we share features of our parents and our grandparents. So these checking of these deviations is at a molecular level by cyclin proteins or cyclin-dependent kinase. So this is about what we call cell division, types of cell divisions, and what we call the regulation of cell division. Now, then we move to what we call the extranuclear genome. Extranuclear genome, that is, I told you, the genetic material or DNA, which controls all the features that take place in an organism, or mostly directly under the control of the nucleus, especially your genetic. See, as I talk, as I prepare, as I present certain features before you, these are under control of my brain. My brain is very active, is guiding me, is controlling my speech, It's controlling my expressions, etc. They are under the control of my brain. So also, if you take a cell, the nucleus is equivalent to that of your brain. All activities that take place in the cell are under the direct control of what we call the nucleus, and especially the chromosome, or more specifically the DNA, still more specifically the gene. Gene is nothing but a part of the DNA, a part of the DNA that controls, that regulates various cell functions is known as a gene. So now generally we have, this is what we call nuclear genetic material, because genetic material is located in the nucleus. But there, not only in the nucleus, but some amount, maybe 1% or less than 1% of the genetic material or DNA is present in the cytoplasm. There are two important organelles where the DNA is present. They are your mitochondria and chloroplast. The DNA of the mitochondria is known as mtDNA. MT means mitochondrial DNA. cpDNA means, CP means chloroplast. DNA of the chloroplast is known as cpDNA. So these two organelles are otherwise known as semi-autonomous organelles. Why they are known as semi-autonomous organelles? These are the only organelles that are present in the cytoplasm. They have their own genetic material. That genetic material is present in what we call matrix. In the matrix of the mitochondrion, in the matrix of the chloroplast, you have a circular naked DNA. A circular naked DNA. Why we call naked? Why use the term naked? Because it is not bounded by any histone. Histone proteins are there. If histone protein is there, then you will see formation of what we call chromosome. But this histone is absent. So that's why you find a naked DNA ring. DNA in the form of a ring. As there are no proteins, that's why naked DNA ring is there. And this is present in the mitochondria and also in the chloroplast. In addition to this, they also have what we call, I told you, like your bacteria, they have what we call their own ribosome, 70S ribosome. Ribosome is the only organelle that is present in all cells. There is no organelle other than your ribosome that is universal in its distribution. But of course, you have 80S ribosome in eukaryotes and 70S ribosomes in your prokaryotes. So here, what I mean to say here is we have what we call the DNA is present in the cytoplasm. That's why it is cytoplasm DNA. It is not, this DNA is not present in the nucleus. That's why it is extranuclear DNA. So extranuclear DNA or cytoplasmic DNA, it is otherwise called, as it is present in the mitochondria, it is known as mitochondrial DNA. As it is present in the chloroplast, it is called. And that's why, because this DNA helps in the duplication of these organelles. It is because of the presence of this DNA, the mitochondria can duplicate, can replicate. So is the case with the chloroplast. But don't forget, they are semi-autonomous. They can't go on reproducing independent of your nuclear DNA. Ultimately, they are under the control of DNA. But a bit, because they have a few number, maybe 100 genes are there. But when nuclear DNA, they are in lakhs. So here they are just in hundreds. So, and even the length what we call here, what we call 16.5. For example, in case of you and me, in case of mammals, the length of this mitochondrial genome or the DNA is just 16.5 kbs, means kilo base pairs. Base pairs means your adenine, guanine, thymine, AT and GC. They are nothing but base pairs. So such base pairs number is kilo base, thousands. So kilo base pairs are there, and it is 16.5 in case of mitochondria, and it is around 120, 150 kbs in case of chloroplast DNA. So they have their DNAs in the matrix. They have their right, because this is there, and they have ribosomes. So these ribosomal DNA, these ribosomal units, to some extent, the ribosomal DNA and the transfer RNA and the ribosomal RNA are produced by the genes that are present in the mitochondria. So mitochondrial RNA, rRNA, mitochondrial tRNA, mitochondrial chloroplast, RNA, chloroplast rRNA, chloroplast tRNA are under the control of these extranuclear DNA that is present in the matrix of these two organelles. But there are certain, I told you, they are just semi-autonomous. And first of all, let us speak about what we call mitochondrial. Here, these mitochondrial, I told you, it is a double-stranded, because it is a DNA, it is a double, two strands are there, but these two strands do differ. They do differ. One has what we call L strand, the other is what we call H strand. H means heavy strand. And a number of genes are more in heavy strand and less in L strand, light strand. And they control, mostly they production of tRNA, production of rRNA are under the control of these mitochondrial DNA. And they too help in what we call oxidative phosphorylation. The mitochondria, otherwise known as powerhouse of a cell. It is involved in what we call, when you speak of what we call respiration, glycolysis, Krebs cycle, and last one is what we call electron transport system, where production of ATP takes place. That stage, what we call oxidative phosphorylation. So some enzymes of oxidative phosphorylation or production of some enzymes are under the control of what we call mitochondrial DNA. Now there are one or two major differences with nuclear DNA and this one. See, to cite, for example, in DNA, generally we have, of course, we learn in our next classes about genetic code. In genetic code, we have seen the genetic code is a sequence of 3 nucleotides. That is what we call a codon, and of course anticodon. Codon on DNA, anticodon on mRNA. So on in DNA, of course, there are codes are there, and for example, there are 3 codes, what we call UAA, UGA, UGA, UAG, UGA. These are known as end codons, or otherwise nonsense codons, because they do not code for any amino acid. So of the 3, generally, if a codon is what we call UGA on the DNA, nuclear DNA, generally it codes for what we call end, nonsense, do not code for any. If the same codon is there on the mitochondrial DNA, it codes for tryptophan. The nonsense codon of nuclear DNA is the sense codon in empty DNA. And there is one more, for example, UGA is there. UGA is the end codon in mitochondrial DNA, but the same AGA, this codes for what we call arginine. Arginine, it codes for arginine in case of AGA, in case of nuclear DNA. The same AGA is the end codon for mitochondrial DNA. Apart from this, male sterility genes of maize are restricted on the, are present on the mitochondrial or DNA. And also in case of Pokey mutation of neurospora, this gene is also located in the mitochondrial DNA. So this is about mitochondrial DNA. Then we move to, for chloroplast DNA. Chloroplast DNAs are a little bit big when compared to mitochondrial DNA. And these, they are similar to mitochondrial DNA. They are there in the matrix, and they have hundreds of these small extranuclear rings are there in the matrix of the chloroplast. And in some cases, for example, in the chloroplast genome, of certain plants has been studied very closely, very in detail. And for example, chloroplast DNA of mirabilis, there are roughly 72 genes are there. Of these 72, generally 45 code for RNA and 27 code for what we call proteins. And some code for what we call RNAs, and some code that is RNA, some code, and tRNA for some genes code, and for some, they code for what we call, G, that is proteins, polypeptides. They code for polypeptides. So here, so cpDNA, that is chloroplast DNA, it codes for what we call some 45 genes code for rRNA, some 20 odd code for what we call tRNA, and 27 code for what we call proteins and enzymes. And in one last thing I want to stress here is, for example, in Mirabilis jalapa, 4 o'clock plant, we have what we call some flowers are white, some are red, and some are, of course, pink. We'll study in our next class. That is dominance, incomplete dominance, codominance, etc. The deviations of Mendelian genetics we'll learn in our next class. So in Mirabilis jalapa, not only flowers, but even you find certain color spots on the leaves. And these colored spots are under the control of the chloroplast DNA. So in the chloroplast, the colored spots that are observed on the leaves of the Mirabilis jalapa are found to be on the leaves. Okay. So now this is about what you see. This comes, mitochondrial DNA or chloroplast DNA comes under what we call only short nodes. So this your meiosis process or mitosis process comes under what we call assay type. There's significance also. You so regarding this chloroplast DNA and mitochondrial DNA, you learn about kilobase space and their matrical presence in matrix, etc., etc. And then you can make your short note very you can score good marks. So with this, what I can say, we of course we had to run very fast because time is very little and the number of classes allotted are very little. So that's why I have to run a bit fast, and we have completed. So like this today, you cell division, types of cell divisions, and the extranuclear DNA. And in next class, of course, as you know, on the coming Saturday and Sunday, we have 2 classes more, and then we'll try to complete the leftover syllabus. So
Okay.
if you have any doubt, Navya, but of course the time is running. But if you have any doubt, I'll try to clear your doubt. If you have no doubt, we will call it a day, and I'll close today's lecture.
No,
Navya?
sir.
Any doubt, Navya?
No, sir.
You are bored because botany ante bore kada. Botany class ante bore. Okay.
నేను బాటనీ ఎక్కువ ఇష్టపడతాను.
Okay. Manchidamma, wish you all the best. If you have any doubt, try to raise them in the next class.